Welcome, everybody. So today we're going to finish up talking about acid-base homeostasis involving the respiratory and renal systems, and then we're going to move on to the GI system. And hopefully some of this new GI stuff will be familiar because you've already seen these organs in lab. Okay, so hopefully you have had a chance to jot down a couple thoughts or look back a little in your notes. So we're trying to think up the ways that the renal system can compensate for respiratory acidosis. Now, hopefully we remember that what this question is really asking us. We're talking about the pH of blood, right? The pH of plasma. We have our two states, acidosis, where our blood is too acidic, right, below 0.35. This is usually going to be due to a buildup of carbon dioxide as a biologic acid, especially when we're talking about respiratory acidosis. So we might expect respiratory acidosis to exist if we're not breathing enough, so carbon dioxide is building up in our blood. And the renal system is one of the ways we could correct for this. So we have various mechanisms for adjusting our pH, kind of in terms of their timelines for how fast they act, right? We can buffer it out with hemoglobin, but especially with bicarbonate. We can use the respiratory system to adjust our breathing. But if the respiratory system is the problem, that's not going to help us. So we move to the renal system. So basically what the renal system will do here, if our plasma is too acidic, which it would be in the case of respiratory acidosis, right? So one thing we can do to bring that pH back up, right, is we can get rid of hydrogen ions. So if we get rid of hydrogen ions, if we excrete them out, they're not in our plasma anymore. So that will pull our pH back up. So the renal system can increase secretion of hydrogen ions in response to acidosis. And we've seen a couple of places where our nephrons secrete hydrogen ions into the tubular fluid, from whence they would then end up in the urine, right? Once they're in the fluid, they continue down through the ureter into the bladder and out of the body. So we can see in the proximal tubule, in the epithelial cells, we had secretion of hydrogen ions coupled to reabsorption of bicarbonate, right, which we can follow vaguely down this kind of pathway. So that's how our bicarbonate traveled. Our hydrogen ions are secreted. So that's one place we see secretion of hydrogen ions by the proximal tubule. We saw in the distal tubule and collecting duct, we had hydrogen ion secretion into the tubular fluid again. This time it was coupled to the creation of new bicarbonate, basically from carbon dioxide, right? So here we are coupling creation of new bicarbonate to hydrogen ion secretion. We actually also increased secretion of hydrogen as we were breaking down glutamine and creating bicarbonate from that. This was also happening in the proximal tubule. So we talked about glutamine as kind of like the last thing that the kidneys would do. It was our last picture of a cell, but this is actually also in the proximal tubule. So we could link these two pictures together. Okay. So that's the secretion of hydrogen ions part. So that would end up with you urinating out hydrogen ions, bringing your pH back up. Okay. At the same time, we would want to increase the reabsorption of bicarbonate because bicarbonate acts like a sponge for hydrogen ions. So if we get bicarbonate in the blood, it's going to soak up all the extra hydrogen ions that are making our blood acidic. So if we can get more bicarbonate into the plasma, that's great. That's going to help us lift that pH back to normal. So while we were secreting hydrogen ions in this proximal tubule epithelial cell, right, we were reabsorbing bicarbonate. So we had a relatively long process for this, right? We took the bicarbonate, turned it into water and carbon dioxide, absorbed the carbon dioxide, turned it back into bicarbonate and hydrogen ions, and then reabsorbed the bicarbonate. But the end result is just reabsorption of that bicarbonate. And in some of our other cells, we're going to be synthesizing new bicarbonate, again, to end up with more bicarbonate in the plasma. So we may be absorbing carbon dioxide and turning it into bicarbonate ions that we pull into the plasma. That's what's happening in our distal tubule and in our collecting ducts, specifically in their intercalated cells. In our last unit, we were talking about the principal cells in the distal tubule and collecting ducts. These are a different type of cell that are responsible for pH regulation. Or we might be creating bicarbonate from glutamine in the proximal tubule, as we see here. Okay. Questions about this before we move to our kind of ending summary slides for our acid-base regulation and then move into the GI? Okay. Oh, great. Yeah, bicarbonate is basically a base. Yep. Yeah. So here we see the Henderson-Hasselbach equation, which we saw before, right? We're not concerned with directly calculating out the pH, although we see that it would be possible if we looked at ratios of bicarbonate to carbon dioxide. We remember that everything we're doing when we're compensating for acid-base disturbances is we're going to be trying to control this ratio, and we want to keep it at a very specific number, right? We want to have our ratio be 20 to 1. So we want to have a lot more bicarbonate than we have carbon dioxide. Okay. So if this ratio gets too low, right, say our bicarbonate went down to 15, so then it was 15 to 1, that would be a problem, right? That would be acidosis. Or flip side, same thing, if we kept it at 20, but our carbon dioxide went up, right, went up to 5, that would be acidosis, right? So anything where this ratio goes down. And the reverse, alkalosis, if we have a decrease in carbon dioxide or an increase in the bicarbonate relatively, we'd end up with a bigger ratio, which would be alkalosis, a pH that is too basic for our regulated range. And so the kidneys and the lungs are regulating basically two different pieces of this. Our kidneys and our renal system are responsible for the bicarbonate part, and our lungs, the respiratory system, are responsible for the carbon dioxide part. So when we pull together these two concepts, we can think about where the problem is and which system is going to fix it, right? Because we can fix this problem by altering either bicarbonate concentrations or carbon dioxide concentrations. So we have two options, and different options are going to be appropriate depending on where in the body we have a problem. Okay. So in the case that we have respiratory acidosis, right? So respiratory acidosis which is actually what we went through with our warm-up problem, right? So respiratory acidosis means we have too much carbon dioxide, basically because we're not breathing enough, right? So if you're breathing too slowly or not breathing, we'd call that hypoventilation. It means carbon dioxide is building up in your blood. Carbon dioxide is a biologic acid. Essentially, it turns into increased hydrogen ions in the plasma. Now, if we are in respiratory acidosis, that means the problem is our lungs. So we can't adjust the carbon dioxide content of the plasma using the lungs because that's where the problem is, right? So we are going to compensate using our other system. So we're going to do renal compensation. So we'll do the things we just talked about, right? We'll secrete more hydrogen ions. So we're going to try to urinate out some of those extra hydrogen ions. And we're going to increase bicarbonate reabsorption and possibly also bicarbonate actual synthesis in order to move more bicarbonate into the plasma in order to soak up those hydrogen ions. So the reverse will happen in respiratory alkalosis, right? So in respiratory alkalosis means we're too basic, means we don't have enough carbon dioxide. And a reason for that could be that we are breathing too much, right? You're huffing and puffing. You're filling your blood with oxygen, but you're depleting all the carbon dioxide. Since we don't have enough carbon dioxide, this actually leads to a decrease in hydrogen ions, right? So overall, we are increasing the pH, making it more basic. The same thing if this is a respiratory problem, our compensation is going to be renal. But now we're going to do the reverse thing, right? So we're not going to increase hydrogen ion secretion. We actually want to stop secreting and therefore excreting hydrogen ions. We want to keep them in the plasma. We want to maintain our pH. So we're going to stop peeing them out in order to try and draw that pH in the plasma back down, right? And same time, we don't want to buffer out so many hydrogen ions, right? We want more hydrogen ions in the blood, which means we don't want as much of our sponge, don't want as much of our bicarbonate. So we are going to decrease, kind of stop our bicarbonate reabsorption and definitely the bicarbonate synthesis. So we can then look at the flip side if we have metabolic causes for our acidosis or alkalosis, respectively. So if we have metabolic acidosis, this still means that our plasma is too acidic, right? So it could still be diagnosed by an increase in hydrogen ions. But if we are in metabolic acidosis, that means that the problem is not the respiratory system, means it's not carbon dioxide that is causing this increase in hydrogen ions. So this is going to be something else, right? So this might be the case, right? We talked about, for example, how when you have diarrhea, right, it actually makes you excrete out a bunch of bicarbonate there, which has the overall effect of increasing hydrogen ions in terms of the balance of your plasma. So a cause like that might be metabolic acidosis, might be one of the reasons for metabolic acidosis. So if that were the case, if that were our specific reason for this metabolic acidosis, we'd be able to compensate using both the respiratory and the renal systems, right? If your problem is your GI system, you can use both the lungs and the kidneys to fix it. So you could compensate using each. You do it in kind of this order. So the respiratory compensation, so you'd start to breathe more, right? You'd breathe more, which means you're breathing off more CO2. So this would bring your CO2 down, which would have the result of adjusting your pH back up, right? Because we're getting rid of this acid. And it doesn't matter that we're breathing off CO2, but our problem is hydrogen ions. We can kind of group those together when we're talking about pH in the plasma. So it's okay that we're talking about different molecules here because they are both acting as acids in our plasma. They can kind of be converted from one to the other. Okay. So in metabolic acidosis, presuming our problem is not the kidney, we can also do renal compensation, which we just discussed as we were talking about respiratory acidosis, right? So renal compensation, compensation for acidosis specifically means we're going to be doing hydrogen ion secretion, we're reabsorbing our bicarbonate, and we are going to synthesize new bicarbonate. In alkalosis, we have the reverse happening. So metabolic alkalosis, a reason for this that is not a renal problem might be like vomiting. So when you vomit, you actually vomit out a bunch of stomach acid. So this decreases the overall acidity. So that would be a cause of alkalosis that is not carbon dioxide. So we could adjust using both respiratory and renal systems. So the respiratory compensation for this would be to increase our CO2. So to breathe faster, or sorry, to breathe slower. The decreased ventilation means you're not breathing off the CO2 as much, which means it's staying in your blood, which means it's going to act as an acid to kind of balance out the fact that we have decreased hydrogen ions. For the renal compensation part, we're just going to flip what we've been doing to adjust for acidosis. So we are going to decrease the hydrogen ion secretion, meaning we're maintaining hydrogen ions in the plasma. We're going to decrease the bicarbonate reabsorption, meaning we are not going to have as much of that bicarbonate buffer in the plasma. Same deal, we are going to decrease the synthesis of new bicarbonate. We're not going to make new bicarbonate because we don't want as much bicarbonate in our blood anyway. So we will practice this later on, basically using this chart. So this is kind of a primer to what we would look at if we're looking at this chart. So this basically summarizes all these different mechanisms we've been talking about. So we start out by measuring our arterial pH, right? So just the pH of plasma. We know we have a specific range, 7.35 to 7.45, that's normal. But if we're beyond that range, we're either in acidosis or when we're in alkalosis, right? So that's the first kind of definitional part that we went over of acidosis versus alkalosis. Now, the next part of this chart is it breaks down how we would identify whether the cause is metabolic or whether the cause is respiratory. So we can determine that at the initial state by measuring the bicarbonate and the CO2, right? So we're doing this because we're trying to figure out whether it's actually carbon dioxide, this gas that acts as a biologic acid, that's the problem. If it's carbon dioxide, if the problem is the carbon dioxide, this means we are in respiratory acidosis. So we compare this to our normal expected PCO2 our PCO2 in arterial blood, which is 40. So we expect the pressure of carbon dioxide to be 40 millimeters of mercury in arterial blood. So if it is not, right, if it's more and we have an acidic pH, if we're in acidosis and our carbon dioxide is above 40, we know that's respiratory acidosis. Okay. So that would diagnose respiratory acidosis. The opposite, if we measured bicarbonate, the sort of threshold, the place we expect our concentration of bicarbonate to be is 24. So if the concentration of bicarbonate is less than 24 millimoles, we would expect that to be metabolic acidosis. Okay, so we would measure these two numbers, but then your body would do compensation, right? It would compensate for these problems, either using the respiratory system if the problem is metabolic, or the renal system if the problem is respiratory. And that is going to result to a second measure. So using the second measure, Sorry, someone just joined us. Let me let them in. Okay. So using this secondary measure, we're going to see that, let's follow this track, right? Okay, so our problem with CO2 was too high, right? So if we measured CO2, maybe it's like 45. This means we're in respiratory acidosis, which means our kidneys are going to compensate. We're going to do renal compensation, right? So we're adjusting our hydrogen ions and we're adjusting our bicarbonate, right? So specifically, we're trying to end up with more bicarbonate in the plasma in order to sort of buffer out the excess acids in the plasma. So if we then measured the bicarbonate at this point, we would see that it is also above 24. So what I am trying to lead you to by walking through this chart, and like I said, we'll practice it later as a kind of review, but taking these three measures, I should say, you can diagnose fully what's happening in the body, right? So if you have a low pH, a high CO2, and a high bicarbonate concentration, that tells you very specifically that you're in respiratory acidosis and you're doing renal compensation. And each of these pathways elsewhere, each of them are identifiable as well by a specific relationship of these three values, the pH, the CO2, and the bicarbonate. So like low pH, low bicarbonate, and low CO2 tells you it's metabolic acidosis doing respiratory compensation. And the same thing would happen on the right-hand side. So by taking just these three measures, you can figure out what's going wrong with the patient, right? And what their body is doing to adjust for it. So this is really, really helpful. You could do this just with a blood draw. You don't have to go look at a kidney. So we will come back to this and review another day, but for now we are going to move on to the GI system so that we can get going on our new chapter. So we're now on to chapter 20. So we're beginning by just identifying the parts of the GI system. So we're going to start with the organs, which you've already seen in lab. And we're going to talk a little bit more in depth about them in terms of their tissue layers, which I don't think you really did talk about in anatomy lab. And from there, we're going to move into talking about the glands and the actual process of digestion of food as it moves through. So our overview here, the reason we're interested in the GI system, right? So when you eat food, right, say you eat a burger, right? The food that you're consuming has nutrients in it, but they are in too large a form to directly enter into the bloodstream, right? You can't transport just a chunk of burger into your plasma because you need protein. It's not going to work like that. You need to digest these things before you absorb them. So we're going to need to do some mechanical digestion of food, so physically chewing, right? But we're also going to need to do chemical digestion of food, breaking down the particles into molecules that are small enough that we can get them into the bloodstream. So all our nutrients are going to undergo digestion, right? Digestion is the breaking down of these food particles and large molecules before we can absorb them. So we're now talking about absorption, not reabsorption. When we're talking about reabsorption, right, in the kidneys, we were talking about something that had been filtered into the tubular fluid and then is going back to the plasma. Now we're talking about absorption. It's just going from your gut into the plasma. There was no initial filtering step, which is why it's a different word, but it's the same direction, into the blood. Okay, so we're going to break stuff down, digest it before we can absorb it or move it into the plasma. So we're going to have two processes that help us with this. So we are going to see motility is a facet of our GI system. So motility is going to refer to basically the movement of our gut. We're also going to see secretion, which we just talked about with hydrogen ions, essentially, right? So secretion, we are going to be secreting or moving particles from the body into the GI tract in order to help us digest those food particles and aid in absorbing them. So we're going to be tracing all these factors as we kind of move through the GI system. So our GI tract can kind of be broken into two. So we can think about the movement of food on its own, and then we can think about the glands that create these secretory products that are going to help us digest the food. So we want to pay careful attention actually to where these two systems, these two divisions of the gastrointestinal system kind of intersect, right? We are going to want to know where these glands are depositing their different enzymes, their different fluids that are going to help us digest food. But we can trace the basic flow of food. Right? We have a hollow tube going from the mouth to the anus. Right? So we have an entrance and we have an exit. And fluid follows a very specific path. So food goes in through your mouth, right? So through the oral cavity, goes into your pharynx or your throat, right? Then travels through the muscular esophagus, which runs behind your trachea, so behind your respiratory system. Travels all the way through the thorax, entering behind the diaphragm into the stomach. In the stomach, we're going to see we have a lot of acids that are going to help kind of kickstart digestion. We're then going to move into the small intestine. And remember, the small intestine has parts, the duodenum, jejunum, and ileum, before we end up in the colon, which has an ascending, a transverse, and a descending part, and then a sigmoid part, before we enter the rectum and leave through the anus. So that is how our food is traveling. We're going to see that we have some very important glands involved with allowing food to be digested in these locations. So we've seen salivary glands in labs. So we're actually going to start digestion way up at the top in the mouth. We're going to see that we have a bunch of glands associated with the stomach, which we'll talk about as we get there. We're also going to see that the liver and the pancreas are very involved with producing these enzymes and fluids that allow us to digest as well. So these different accessory glands are also considered part of our gastrointestinal system. So we're going to start by looking at basically a cross-section of a wall of the GI tract. So we have some basic kind of cell layers that we're interested in as we're talking about the GI tract. So there are basically 4 of them. And these are going from inside. So the inside of the GI tract, just a reminder, is called the lumen. So the place that your food is passing is the lumen. And we will often refer to the food and fluids that are in the GI tract in the center hollow area as luminal contents. Just means the stuff that's inside this hollow cavity inside the GI tract. Okay, so in the wall of the GI tract, we start out with the lining of the lumen. So the lining of the lumen is called the mucosa. So here we have the mucosa, and it's going to have several parts, which I believe is going to be our next couple slides. So we won't talk about them yet, but the mucosa is going to have its own sublayers basically. But outside the mucosa, we're then going to have a layer of connective tissue called the submucosa. So this is the submucosa, is this purple part basically. Then we have the Muscularis externa, which is a relatively large layer, large for the gut, of muscle, kind of on the outside of our GI tract. but it's covered by one final layer called the serosa. The serosa, here's what you would have actually been touching in anatomy lab, right? That slimy connective tissue on the outside is the serosa. So those are our 4 layers, and they each have kind of different jobs that we care about. So we're going to run through those each in a little more detail. Okay. So we're beginning with the mucosa. So the main role of the mucosa, right, is to separate what's in your GI tract from the rest of your body. So the first part of the mucosa is going to be a mucous membrane consisting of different types of epithelial cells. So we're talking, the mucous membrane is like the first layer of the mucosa. So I've numbered it 1A. We're talking about like these cells are our mucous membrane. Right, so this cell layer. Okay. So these are all epithelial cells. So they're creating a physical barrier between the food in our GI tract and the contents of our body. They're epithelial cells. They come in basically 3 types. So they have like 3 kind of specializations for these epithelial cells. Our absorptive cells, right? Absorptive cells are going to be specialized for helping us absorb nutrients from the contents of the GI tract, so from the luminal contents. Then we're going to have exocrine cells that are going to secrete products basically into the GI tract. And then we are going to see that we have endocrine cells, which do some hormonal signaling, right? Put signals into the bloodstream. So specialized for endocrine function, some of which is related to things like hunger, right? There are signals going to your brain. So some of that is coming from these epithelial cells in the mucous membrane. So our word for all 3 of these different types of epithelial cells together are enterocytes. You're going to see this prefix a lot as relates to the GI system. And we are in a GI unit now, so probably don't have to guess system too much when you look at terms for this class right now. But if in the future you are trying to guess medical terms, if you see this prefix enter, that's usually going to be related to something in your gut. So in this case, we're talking about epithelial cells lining the gut, but we'll also see that there's, for example, an enteric nervous system, which refers to a nervous system in the gut related to the GI tract. So that's a prefix you'll see multiple times here. Okay. So next we're going to go into the other 3 linings. So what the mucous membrane is basically this first thing, the epithelial lining. So our next 2 slides are on the Laminopropia, which is our next layer out, still within the mucosa. And then we're going to talk about the Muscularis mucosa, which is a very thin layer of smooth muscle that you're going to want to distinguish from the Muscularis externa. So we're going to have a very thin layer of muscle here before we get to our thicker layer of muscle here. Okay. So the Laminopropia is what's lying directly outside our mucous membrane. All right, so a little superficial to it. So if this was our mucous membrane with our enterocytes, our specialized epithelial cells, then this yellow area, which I'm going to just kind of squiggle on in blue, right? So this is our Laminopropia. Now the Laminopropia is basically just a connective tissue layer. So it is going to contain small blood vessels and lymphatic vessels. So you can see some lymph nodes actually indicated here, right? Green is your textbook's color for lymph. So we see some lymph nodes. There are going to be some nerves traveling throughout this area as well. But a lot of what we're interested in the Laminopropia, or you will be interested in the future, is this lymphoid tissue, this lymph function. So we have some lymph nodules. We have some specific lymph structures called Peyer's patches here in the Laminopropia. Okay. Mainly think about it as connective tissue. So our third part of the mucosa, is our thin layer of muscle. So our muscularis mucosae. So remember it because it has this mucosae in there, that tells you it's part of our mucosa, which is our kind of bigger term, our bigger layer. Okay. So it's located about here, right? So it's this kind of pink layer that we see. Right, so it's made up of longitudinal fibers and circular fibers. So we have some fibers that are going like all the way around, right, like this. So they can squeeze and narrow the tube. And we also have some longitudinal fibers, right? So we have some longitudinal fibers that would run like this, right? Which are going to be able to essentially kind of lengthen and shorten the tube very slightly. Most of the movement we're going to see in our gut is going to come from our larger layer of muscle, our muscularis externa, which has a similar arrangement of fibers. But this thin layer, this muscularis mucosae, functions in helping to just kind of jostle around the food in the lumen. So it has a little bit of function in basically allowing your food and any acids you're secreting, any other molecules you're secreting to aid in digestion, allows them to kind of mix together in the lumen. So that's kind of its role here. Okay. So all of those three were part of our mucus layer, our mucosa. So now we're moving to the submucosa. So the submucosa is basically this purple layer, right? So we're talking about this stuff. So this is a pretty thick layer, and it's made of connective tissue. So in that way, it's kind of similar to the Laminopropia. But the role of the submucosa, rather than for like lymph function, is really to aid in the physical structure of the gut. So it's really important that as you eat food, right, that pieces of your GI tract can expand as you send a bolus of food through. A bolus basically means, it's a scientific term for like big lump. So as food goes through, you want the GI tract to be able to compensate for that. You want it to be able to stretch out. So that's its distensibility. You also want it to be able to rebound after that food passes through. So that's its elasticity, like we've talked about with blood vessels. So it can be distended, so it can expand out to allow things to pass. And then it can contract back through some elastic properties. And so these two properties are basically properties of this layer of connective tissue, the submucosa. It does have large blood vessels and lymphatic vessels passing through it. So now we can see those here. So we have some large vessels. The lymph part isn't drawn on here on this diagram, but there are also lymphatic vessels here as well. Here we start to have part of the nervous system of the gut, part of the enteric nervous system. So we had some like little small nerves before, but now we're talking about parts of the enteric nervous system. So part of the actual kind of control of the gut. So specifically the part of the enteric nervous system that is part of the submucosa is called Meissner's plexus. And so this is going to be one of the two halves of a nervous system that we can think of as like producing intrinsic gut function. So not requiring stimulus from your brain, really kind of a local nervous system. The second part of the nervous system is in the muscularis externa. So we're just going to run down here to the bottom first. All right, so Meissner's plexus was in the submucosa. So that was half of our enteric nervous system. And Auerbach's plexus, or the myenteric plexus, is the other half of our enteric nervous system, and it's in the muscularis externa. Okay. So this is, again, adding into our intrinsic control of the gut. Physically, the muscularis externa is made of 2 layers of smooth muscle, just kind of like our layer of muscle in the mucosa, just like that little bitty layer. It's made of an inner circular layer, which allows us to change diameter, right? So kind of constricting and narrowing versus expanding and dilating. An outer longitudinal layer that changes length. And when we talk about motility, which will be a little bit later on, when we talk about how food kind of gets squeezed through the gut and jumbled around, we'll think a little bit more about these different layers of muscles and how they function. But that's where this main movement of food through the gut is coming from, the muscularis externa. Okay. So our final layer of the GI tract is the serosa. It has an inner layer and an outer layer. So there's an inner layer of connective tissue, so just normal connective tissue like you saw in phys lab, and an outer layer of something called mesothelium, which is a modified epithelial tissue. Basically, mesothelium is that slimy stuff that you were touching in lab, and it's continuous with the mesenteries. So the mesenteries were those structures that we saw that kind of anchored the GI tract to the posterior wall of the abdomen, which we call the peritoneum, right? So the outer layer of the GI tract itself is made of the same type of tissue that kind of continues and forms the mesentery. So here's our mesothelium, just continuing down. And here you might have the peritoneum, right, the posterior wall of the abdomen. So that's all the same sort of tissue. Okay. Questions about these layers before we then move a little more through the pathway of food? Okay. So our digestion actually begins in the mouth. So our mouth is responsible for allowing us to eat and swallow food, and that eating, that chewing function is actually part of digestion. So that's like mechanical digestion, and we give it a special name, mastication. So mastication is just chomping down, chewing. Okay, so that begins in your mouth with your teeth. And in the mouth, we actually also even start some of our chemical digestion. So we have enzymes that are going to start to break down some of the larger molecules in the food immediately in the mouth. So that's the role of the saliva glands, right? So you know that we have some glands, right? So put our submandibular, sublingual, like our parotid, right? We know there are saliva glands in the head. They're giving saliva into the mouth. So that's how we end up with saliva products there. So in that saliva, we have a fluid that acts as a lubricant, right? It allows us to physically move the fluid around, allows us to kind of not scratch our GI tract up as we swallow food, sorry. But it also contains an enzyme that allows us to start to digest specifically carbohydrates. So in the saliva, we have an enzyme called salivary amylase, right? Amylase digests starch, which is basically a storage polysaccharide, a big carbohydrate, and glycogen, which we know is basically a bunch of glucose molecules linked together, right? We saw it stored in the liver and in the muscle when we talked way back about metabolism in P1, but it's also a big carbohydrate basically. So amylase can also help us begin our digestion of glycogen in the mouth as well if we're eating like animal products. So from the mouth, we pass into the pharynx, right? So the pharynx, basically just conventionally referred to as the throat, right? So the pharynx here is a common passageway for air and food until we hit the region of the epiglottis, where we will see a separation between our larynx and then our respiratory pathways to the anterior portion, and our esophagus, and then the rest of our GI posteriorly. So now we are looking at a sort of longer picture of that esophagus, right? So the esophagus is running basically like here. So we're running from the end of the pharynx all the way down to the stomach. And the esophagus is made of both skeletal and smooth muscle. So some of the function of your throat and of your esophagus can be under conscious control, right? That's true of skeletal muscle. But a lot of the function is going to be under involuntary control, right? You don't think about, if you take a big pill in the morning, you don't have to think for the next half hour about like, am I squeezing my esophagus enough to get it down to my stomach? No, we have smooth muscle that is responsible for the contractions farther down. So we can think about this separation kind of by thinking about the two sphincters on either end of the esophagus. So we have an esophageal sphincter at the top and at the bottom. So we have an upper esophageal sphincter up here, and then this would be our lower esophageal sphincter down here. Lower esophageal sphincter. Okay. So the top esophageal sphincter, the upper esophageal sphincter, can be under conscious control. So it's made of skeletal muscle, right? You can deliberately swallow something. And it's located between the pharynx and the esophagus. So this upper part, directly following the upper esophageal sphincter, which is skeletal muscle, this is the part of the esophagus that is still skeletal muscle. So it's about a third of the esophagus. This top part is skeletal muscle. But then as you move farther down, we turn to smooth muscle, meaning this is going to be involuntary. And the lower esophageal sphincter is made of smooth muscle. So fortunately, right, the movement of food directly into our stomach, that is under unconscious control. Doesn't mean it can't go wrong. We do sometimes have things come back up from the stomach and into the esophagus. This is bad. The esophageal sphincter should not allow this to happen, right? It should be blocking off contents from reversing direction. But that's basically what's happening when like gastric reflux or heartburn happens. So we're having acidic contents of the stomach pop back up through that lower esophageal sphincter. Okay. So that leads us to the stomach itself. So the stomach is located right below the lower esophageal sphincter, and its role is mainly to store food. So it can be very small if it's empty, 50 milliliters approximately, but it can expand a lot. A full stomach is often roughly about 1,000 milliliters. So it's important that the stomach be able to change size so that after you've eaten a meal, it can hold the contents. But if you haven't eaten for 8 hours, it doesn't take up a ton of room, right? And the reason it can do this is because the walls of the stomach have these folds, as you can see. And if you looked in lab, or if you go to review hours, or if your lab is upcoming, you can take a look. They're basically these folds in the walls of the stomach that act kind of like pleats, right? Think of it as like an accordion, right? So the stomach can be small, those folds collapse. When the stomach expands, those folds flatten out, allowing the stomach to get a lot bigger. Now, in the stomach, when we kick off next time, we're going to talk about gastric juices and the different products that help digestion in the stomach. So we're going to see that the stomach is going to regularly contract. So we're going to see that there's a muscular role of the stomach to mix together the food, right, and the gastric juices and the enzymes that we're going to put into it. They're going to produce something called chyme. So chyme is a word we use for this mixture of food and gastric juice, which we're then going to pass into the small intestine through the pylorus. Okay. So we'll leave that here, and next time we'll begin by thinking a little bit more in detail about those gastric juices, the enzymes they contain, and so on. All right. Have a good day, everybody.